All right, for today's class, we are covering basic inheritance, also known as Mendelian inheritance. So this farmer is my Gregor Mendel, and Mendel is referred to as the father of genetics. Even though his training was in theology and mathematics. So he didn't actually discover hereditary material, he just discovered the rules by which hereditary material passes from one generation to the next. And so we call those Mendelian rules of inheritance. And for those rules to apply, we have to adhere to a certain set of conditions. And those conditions arise from Mendel's study organism, which was the garden pea. So there's no garden pea in Minecraft, so I have a few different characteristics to represent some of the traits that he looked at. And so he looked at flower color, so flower colors were either purple or white. Pea color was either green or yellow, so we're going to use an orange pumpkin to represent yellow. And then the plants were either short or tall, so I have a short bamboo stalk or a tall bamboo stalk. And for all of those characteristics, they are discrete. That is to say that you're either purple or you're white. You're not shades of pink. Either you're green or you're yellow. So they're qualitative traits. Discrete traits are also known as qualitative traits. And those traits have two different forms, purple or white, yellow or green. So that's one of the other rules. We can only have two different forms, and Mendel didn't know what those forms were, but we know them now as alleles. And each trait is controlled by one gene. So one gene with two different forms. So right, he didn't know what those were, but he developed the rules that tell us how those things operate. So I said that we needed to understand the cell cycle in order to understand how we get to Mendelian inheritance. And so I wanted to take some of our work from last week and expand upon that. So as an example of what I mean by discrete versus continuous versus non-discrete, so discrete would be either you're white or you're red. So there's a white allele for sheep coat color, or a red allele. And continuous would be that you have shades, you have various forms in between. So we know that most traits are continuous or quantitative, but in order for us to follow Mendelian rules, we have to use traits that are discrete. All right, so Mendel's two rules that will always apply as long as we follow the conditions that we just set forth are the law of segregation and the law of independent assortment. And the law of segregation says that for any one trait, in this case sheep coat color, at the end of meiosis, when gametes are produced, each gamete will either have one form of the trait or the other form, but it'll never have both. That is to say, when we reach the end of the cell cycle, each of our gametes should either be white or red. None of them should be neither white nor red, and none of them should be both white and red. That is our law of segregation, or our law of allelic segregation. And so we go through the cell cycle, and we come to the end where we have our gametes, and what we see is that even after crossing over, which we showed last week, each of our four gametes is either white or red. So that's a visual example of what we mean when we talk about the principle of segregation. And then our other rule is called the law of independent assortment. And so our law of independent assortment says that, we have more than one trait, more than one trait that we can look at any one time. So the three examples that we used were flower color, pea color, and plant height. And so, Mendel was looking at least two different traits every time he did those crosses. So in our example, we'll do sheep coat color and we'll do sheep eye color. And so sheep eye color is either going to be brown or blue. And so I'm going to say that the top half of these chromosomes, this top half here, this orange codes for brown eyes, and this top half here, blue, codes for blue eyes. So the law of independent assortment means that the inheritance of a form of one trait, like red coat color or white coat color, does not influence the inheritance of another trait. So, put more simply, if you end up with red fur color, you could either have brown eyes or blue eyes, and same for white fur color. And we could reciprocate, and we could say if you have blue eyes, you could either have red coat color or white coat color. That's his law of independent assortment, that these traits segregate independently of each other. So it kind of builds upon the first law of segregation. So we're going to take that through, and in independent assortment, we've already talked about what that means is that our chromosomes are swap places with respect to each other when they line up in the metaphase plate. So we didn't show independent assortment here. We go all the way through to our gametes at the end, and remember I said it was coded for by the top half of the chromosomes. So these leftmost two individuals would have brown eyes, but one is red and one is white. So that means you could be red with brown eyes or white with brown eyes. Same for this. Blue eyes and red or white. So that's the idea of independent assortment, that any one trait doesn't affect another trait. And as we move forward and with other material, we'll discover that that's usually not the case, but as long as we're following Mendel's rules, then that will be the case. And then the last thing that I want to talk about before we get into our notability terms is that Mendel, to do his crosses, he had what were called pure breeding individuals. So an individual that was uniform for yellow peas or uniform for green peas. And what that means for Mendel is that if you're a yellow individual and you self-reproduce, which you can do with plants, that you should have only yellow offspring. Same with green. So in his crosses, his initial pea generation were always pure breeding for particular traits. And we use the word monohybrid and dihybrid to refer to the number of genes that we're looking at with any particular cross. A monohybrid cross is breeding two pure breeding individuals that are different from each other at that trait. So a yellow pea individual crossed with a green pea individual would be a monohybrid cross. And then we would follow that to the F1 and follow that to the F2 to get our ratio. And the ratio that we see would demonstrate the mode of inheritance that we have. If we looked at two traits at once, then, like say pea color and flower color, then we would call that a dihybrid cross. Each of our parents would still be pure breeding, but they'd be pure breeding for opposite traits. So one would be yellow peas with purple flowers, and the other would be green peas with white flowers. So one of the problems that Mendel had was that he didn't, just looking at an individual, if he had a pea plant that had purple flowers, he would have no way of knowing if that individual was pure breeding or not. He would have to do what's called a test cross. And a test cross is taking any individual who has a dominant phenotype. So I guess I hadn't hit that, but dominant means that we have these two forms of these alleles, so purple flower color and white flower color. And purple is dominant to white, which means that you only need one copy of the purple allele to be purple, but you need two copies of the white allele to be white. So all of our Mendelian traits show that dominant recessive relationship. So we know that yellow peas are dominant to green peas, and so if we have a yellow individual, then that individual could either be homozygous, have two copies of the same allele, or they could be heterozygous. They could have one copy of each allele. And a test cross is crossing that dominant phenotype individual with a homozygous recessive individual. So an individual with two copies of the green allele, green pea allele. So if the individual in question, the individual on the left side of our Punnett square here, is homozygous, then when we do that cross, all of the offspring will show the dominant phenotype. So all of our offspring here are heterozygotes. They all have one copy of the yellow allele, which means they will all have the yellow phenotype. On the other hand, if my dominant individual in question is a heterozygote, then when I do my test cross it with a homozygous recessive individual, half of my offspring will have the recessive trait, green peas, and the other half will be heterozygotes. Because they have one copy of yellow, they'll have yellow peas. So in this way, Mendel could distinguish among his potential pea plants and figure out which ones he wanted to use in his monohybrid and his dihybrid crosses. Alright, so that brings us to basic inheritance, and I want to run through some of the additional terms that we have in relation to how we're going to understand these monohybrid and dihybrid crosses. So in Canvas we have a full list of all the important terms, but I've kind of picked out some that are most important. And so just as a review, we said that Mendelian inheritance means that we have one gene and that gene has two alleles, and those two alleles have a dominant recessive relationship. So if you're dominant, you need one copy of the allele to show the phenotype. If you're recessive, you need two. Yellow peas are dominant to green peas, purple flowers are dominant to white flowers, and tall plants are dominant to short plants. And then we have our two Mendelian rules. So the law of independent segregation, which means that for any two forms of a gene that an individual has, when that individual produces gametes, each gamete will only have one of those two forms. They separate from each other. And that gives rise to this 3-to-1 ratio. So we have some key characteristics to determine these rules for inheritance, which is why Mendel's mathematical background was so important. And so what he would do is say, okay, if I have a simple dominant recessive relationship, and I have, actually I'm going to back up, I'm going to do a monohybrid cross. I'm going to say A equals yellow peas and little a equals green peas. So my monohybrid cross would be AA crossed with AA, and that's my parental generation. And my F1, so I'll do my Punnett square, but the results should be obvious. All of my offspring are heterozygous. And so let's say I'm going to write these down. So this is a yellow individual, and this is green, and this would be yellow. And then I self my F1, which we can do with plants, we can't do with animals. So to self means you take pollen from one individual and then you cross it with an egg, an ovule from that exact same individual. And that gives us our F2. So our F2 is 3/4 having at least one dominant A, so they're 3/4 A, which is yellow, and they're 1/4 little a, which is green. So that is our 3-to-1 ratio. So what Mendel said was that if we're following all of the rules, all of the basic inheritance rules, and we take a pure breeding monohybrid cross and we take it out to the F2, then we should see a 3-to-1 ratio. We've supported the hypothesis that we have one gene that has two forms of the allele, and one of those is dominant to the other. Right? That's our mode of inheritance. And so he was a mathematician, and so he supported all of these hypotheses with chi-square tests. Right, we should see a 3-to-1 ratio, and he would breed hundreds of plants, and he would get hundreds of offspring. And so he could say, all right, if I have 400 offspring, then I should see 300 yellow and 100 green. And so he could do a chi-square test to determine if his observation was significantly different from his expectation. So our observation minus our expectation squared divided by our expectation. And that gives us our chi-squared value, and so we know if that's larger than our test value, then our difference is significant, and if it's not, then our difference is not significant. So as long as he didn't reject his null hypothesis, and as long as he could say that he didn't have any evidence that they were different from each other, then he could say that they follow those basic Mendelian rules of inheritance. And then our other law is the law of independent assortment, and that gives rise to this 9-to-3 to 3-to-1 ratio. And so like we said in Minecraft, that just means that we're looking at more than one trait. And I know for a lot of people, we'll say that A equals yellow and A equals green, and then B equals purple, and little b equals white. So we'll do a cross, a dihybrid cross involving pea color and flower color. And so, you know, sometimes in high school or in Gen Bio, you learn to make one giant Punnett square. But we're going to be doing a lot of things this semester involving more than 2 genes, and so that gets out of hand really fast. So I would encourage us to focus on one gene at a time and do one Punnett square at a time, and then we can combine the results from those 2 Punnett squares in different ways depending on what our question is. So I'm going to do my parental cross. So I'll just say that my parents are AABB crossed with AABB. So that would be yellow and purple, and that would be green and white. So my parental generation is AA crossed with AA at one locus, and BB crossed with little b at the other locus. Right? Even though each individual has 2 separate genes, I'm teasing them apart because it's much easier to work with one gene at a time. So just like above in my F1, that's going to give me all heterozygotes. And then when I self my F1 to get my F2, just like I did above, the same thing happens. So I'm going to get 3/4 that are A. And so I'm using this underscore right here to represent that could be big B, or it could be big B, little b. So this is a phenotypic notation. I'm just saying that they have the dominant phenotype by saying they're A underscore. So they're yellow and green, purple, and white. All right, so I've teased them apart to make this part easier to get my F2 results per gene. But each individual has both traits, so now I have to bring them back together. And so if I have 2 traits and each of those has 2 possibilities, then that means that I have 4 2-gene possibilities. So I could be big A, big B, big A, little b, little a, big B, or little a, little b. And in terms of phenotype, that would be an individual with yellow and purple, yellow and white, green and purple, and green and white. And so 2 genes, two phenotypes per gene, we always have those 4 possibilities. We always have each of the pairwise combinations of those 2 forms of the phenotype. And, you know, we may or may not see them, but they're all possible. So we want to make sure that we start with all of the possibilities. All right, so now this is the part where the way that you did it in high school, you'd have one giant Punnett square and you just count the number of squares that were each of these things. But what we're going to use is the product rule. So we're saying that this individual is both yellow and purple, yellow and white, green and purple, green and white. So we can use the product rule. We can use their ratio at one locus and multiply by the ratio at the other locus. So big A would be 3/4, and big B is also 3/4. So 3/4 times 3/4 equals 9/16. Big A, once again, is 3/4. And little b is 1/4. So that equals 3/16. Little a is 1/4. Big B is 3/4. So that's 3/16. And little a is 1/4. And little b is also 1/4. So that equals 1/16. So now we have our 9 to 3 to 3 to 1 ratio. So in terms of what Mendel said, it was, if we're just looking at one gene, then we can support the law of segregation. We can say, have we supported the law of segregation? Do we see statistical evidence that we have a 3 to 1 ratio? And then if we see a 9 to 3 to 3 to 1 ratio, right, we'd also do a chi-squared in the exact same way. Do we have statistical support that my traits obey the law of independent assortment? And if they do, then that means I can conclude that their mode of inheritance follows my rules of inheritance. So these set the stage, right? These two tests set the stage for understanding how alleles and genes, the information that they code, passes from one generation to the next. And it's a baseline for things that become more complex. Because a lot of times we're going to do these tests and our chi-squared is going to be significant, which means that something else is going on, which is fine, right? That's the basis of more complex studies of heredity. So before we finish up, I wanted to make sure that I came back and talked about some of the terms that I know that I mentioned without explaining what they were. And so one of those is a locus. And so a locus just refers to a particular location on a chromosome, a specific place. And most of the time when we have one locus, we have one gene. Not always, but most of the time we have one locus and one gene. And so a gene is a region of DNA that makes one particular phenotype. So in our examples, we would have a gene for pea color, we would have a gene for flower color, and we would have a gene for plant height, right? If you have a phenotype that you can discretely describe, then that discrete phenotype has a gene, right? And this is Mendelian. This is basic, right? We know that oftentimes these rules don't apply, but this is our starting point. And then, as we said up above, for each gene, Mendelian rules, we have 2 alleles, at least 2 alleles, and those 2 alleles are the forms of that gene. So white or purple for flower color. And then some other terms that we used were homozygous and heterozygous. And so if I'm homozygous, I have 2 of the same allele at a gene, and if I'm heterozygous, I have 2 different forms of that gene. And then I feel like we've covered dominant recessive pretty well. If you're dominant, you just need 1 copy. If you're recessive, you need 2 copies to show that phenotype. All right. And then in Minecraft, we talked about how Mendel wanted to do pure breeding crosses, but in order to do his pure breeding crosses, he had to do test crosses to determine who's homozygous and who's heterozygous. But once he did that, then he could do monohybrid crosses to look at the law of segregation, and he could do dihybrid crosses to look for the law of independent assortment. All right, so that's it for this week. Thanks very much, and hopefully we'll see you next week.